All right. Hey guys, welcome to tonight's review session. It's for week 4, the second half of it. It's going to be talking about non-coding RNA and the different pathways for RNA interference.
Okay.
That's
Yeah,
just that's just a title slide.
that's fine. Oh yeah, so I'll do the first half on just non-coding RNA and then risk, and then Tina we'll do the different types of SI, MI, and all that.
Yep.
Okay, so non-coding RNA is pretty much just RNA molecules that do not code for proteins, so there's no translation. Um, they're pretty much good as they are because they perform their own functions, which are pretty much just either catalyzing other chemical reactions. So you're just working as um, think of like enzymes. It's just you know how they do reactions like that in a similar sense, and they also regulate gene expression. So they'll be able to splice DNA or promote it or silence it, suppress it, stuff like that. So if you go to the next slide, we can. Oh yeah. So then the types of non-coding RNA, two main ones you really want to know are your rRNA or ribosomal RNA, and those are the ones that are going to be doing the catalytic part of ribosomes, right? They're going to be forming those peptide bonds for protein synthesis. And then you have your tRNA, your transfer RNA, and these are the ones, as you can see here on the figure, they're going to be bringing over your amino acids um, to the ribosome during translation. So you're going to be using both your rRNAs and your tRNAs when you want to translate mRNA to a protein. And yeah, we can say
Oh,
something.
sorry. I was just going to add, I know that sometimes some people get confused. So I thought it would be helpful to say that like the ribosomes, like Saya just said, the rRNAs are the catalytic part of the ribosome. So like this entire ribosome is made of proteins and RNA. And then it's just that the rRNA are the catalytic parts of it. So they're the one that's like forming these bonds, the bonds to form the amino acid chain.
Yeah. I should be more specific with that. The RNAs are specifically those parts that will be putting those bonds together. Um, but yeah, and tRNAs, the other thing is that they're going to be important for base matching. So either it's going to be 100% match or maybe close to 100 depending on the situation. But that's how the cell knows which amino acids are going to be going on to the codons on the mRNA. But yeah, that's pretty much that there. And then other notable RNAs, they're good to know, but I don't think they're going to be a big focus of the exam at all. But first we have snRNAs or small nuclear, and those are the catalytic part of spliceosomes. So just like um, the RNAs before on the last slide, the rNRNAs, they're going to be ribozymes, so right, like enzyme kind of RNAs. And then you have your long coding RNAs, and these are going to be, um, for the purpose of this class, you're just going to need to know that they're involved in X chromosome inactivation, right? You have exist and its reverse T6, and pretty much your long coding RNAs. Think of it as two little, I guess, brackets. You can think of the shape. They're gonna come and they're gonna pick one of the X chromosomes, and they're just gonna go around that X's and T6 and kind of inactivate it that way. I don't think you really need to know the specifics of that mechanism, but that's just the function. And then you have CRISPR RNA. This is going to be your DNA suppression in bacteria or prokaryotes, I should say. Um, and pretty much it works by using guide strands that it saves from, like, say, if a bacterial cell gets infected by a virus, it's going to save some of that DNA. So for next time when it does, the CRISPR part will be able to look through that catalog of stored DNA from exogenous sources. It's going to find that invading virus, and then it's going to know by matching it to the infecting virus, it's going to be able to know that, or this is the part of the DNA that I need to cleave by DNA endonucleases. So it's basically, just to sum up, it's a way for bacteria to kind of like you think of our antibodies, right? Like say you have antibodies, your cell is then prepared to know, to recognize a foreign invader when it comes again to then cleave that DNA. Um, and then you have SRPs. This one's probably pretty important to know because it has to do with protein translocation. It's pretty much just a ribonucleoprotein, right? So it's a ribosome, nucleo, and then a protein part that is just used to translocate proteins into the ER. And then lastly, you have your RNases. And these are just RNAs that are used to cut other RNAs, ribonucleoproteins, in where the RNA portion or the RNA part is the catalytic part. So that's going to be your enzyme analog, right? So that's going to be, again, going to be cutting bonds or making bonds. But yeah, that's pretty much it for that.
Sorry. Okay.
Fun.
Just like a little stop. Any questions? Or I guess specifically Haley, if you have any questions.
Yeah, that was a lot. So if you have,
None so far. I kind of understand the non-coding RNA. So
All right, cool.
yeah,
So
those are, you just pretty much need to memorize what they do. Nothing like, I don't think it's going to be anything in
yeah,
depth.
exactly. I think I just put them in because in Laura's videos she mentioned them. So I thought they'd be like useful to know, and it just kind of shows like in general that RNA isn't just always used to make proteins. That there's a lot of use for different types of RNAs like within the cell too.
Yeah, exactly.
yeah, it also like contextualizes like SRP too, just
Yeah.
in like the stuff we learned before. So
Yeah, that's definitely useful from last week. Like the translocation to like the ER. That's all with the SRP that does that.
Yeah. So, and then next we're going to go to RNAi, which is basically RNA inactivation. So pretty much this is this is going to be found in your eukaryotic cells. It's going to be this part's important to remember. It's going to be your post-transcriptional control, right? You have different types of mRNA control, right? Your post-translational, translational. So you're going to want to know that this is going to be for your post-transcriptional mRNA. So you already have the mRNA from the DNA. Now you're going to further modify it at this step. And pretty much for this, you're just going to be using it for gene suppression. And it's a big focus in research and therapeutics because one of the best ways to know what a protein or DNA or gene does is to, knock out. So let's say we have a gene or a piece of mRNA. If we inactivate it and then we study the downstream effects, or we study what results of that, then we know that, okay, now we know for certain that this piece did this part. So that's why it's like really good for research. And then pretty much the thing we want to focus on for RNAi here is the risk complex. That's what's going to be doing the main interference machine, right? So it's going to be RNA, it's going to be argonauts protein. It's a complex of that. And the two pieces of RNA that's going to be working with are your microRNAs, which is for your endogenous gene suppression or silencing. And then you're going to have your siRNA, which is going to be your exogenous. So think of a foreign invader such as a bacteria or a virus. I got an email actually today about siRNA. Somebody was asking, does it also involve, like say, something invading from like another part of the organism, like another cell in the organism? And I guess technically it could, but I've never really. The main focus is usually just another virus or a bacteria. It's usually not to do. It doesn't usually have to do with like say another cell within the same organism. So I thought that would be a good thing to clarify if anyone else has kind of question. Yeah, more on the risk complex. Pretty much, yeah. Again, it has to deal with mRNA and siRNA pathways. The two pathways are very similar. It's just their starting point is different, and we'll go into that later. But pretty much mRNA, think of inside the cell. Say a cell is producing too much of an mRNA or has too much of a protein, right? The cell doesn't want to waste any energy on making stuff that it doesn't need to, so it'll just, use mRNA to kind of sequester that. And then you have siRNA again. Um, Say there's a foreign invader, the cell is just going to flat out degrade that. So it doesn't need to sequester it like it would for miRNA. It would just take that exogenous invader and then just mark it for ubiquitin degradation. And so different organisms use si and mRNA in different ways. But for this class, you'll just need to know how it works in humans. And pretty much the general mechanism is that you're going to get your RNA, either it's MI or SI, and in the RIS complex is going to unwind the two strands, right? Because it's going to come to it as a double-stranded RNA, right? So yeah, the RIS complex is going to take the two double-stranded RNA, it's going to unwind it, and then it's going to pick one of the two to use as a base pair, to then use that to then match it to whatever it needs to suppress. So right, it's easier for it to be mRNA, right? Because it's endogenous, it already has it's the same cell itself. So it doesn't need to be a perfect match for that. But for siRNA, since it's a foreign invader, it's gonna usually always be a perfect match. And yeah, pretty much that just results in either translation or degradation, or regulating translation or flat out degrading the gene.
Yeah, so before we go into like the more specifics of miRNA and siRNA, does anyone have questions so far? Anything they want to like more clarify or anything? All right, if not, we can just keep going. So a little bit more about micro RNA. It's a post-transcriptional regulator. Like Saya just said, it acts as a guide strand for RISC. So the miRNA RISC will use that one of the strands that it chooses in order to target and base pair with mRNA. And then because of that, it'll regulate the mRNA stability and localization. And then usually microRNAs are very small. They're about like 20 to 25 base pairs. I totally spelled that wrong, but they're basically also double-stranded RNA. And then, like Syed said as well, they're endogenously expressed, so encoded for within the genome. So they're expressed in the nucleus. It's part of the DNA genes that the cell has in itself. It doesn't have to come from somewhere else outside. And it's actually not incredibly useful or prevalent in humans. They regulate less than 60% of the human's protein coding genes. So it's there and it definitely is useful, but it's just not used as much as say like siRNA, I believe. So for the mRNA pathway, this kind of helps visualize it a little more. So first you start off with RNA polymerase II. It will go and transcribe the initial transcript. So that initial transcript, it's called pri-mRNA. So it's still not pre-miRNA yet. It's just like the very basic initial transcript, like nothing's been done to it yet. It's just initially transcribed. And then once you transcribe the pri-mRNA, it goes into the first cut processing to become pre-miRNA. And this is done by drosha, which is a RNA nuclease, which is something that we talked about in those first few slides about other non-coding RNAs. It's just a nuclease where it can it has the ability to cut and have that enzymatic or like catalytic ability to cut the RNA pieces. And so once drosha goes through processes and cuts the RNA, the pre-miRNA is now exported out from the nucleus into the cytosol. And then this is just kind of important to know because we learned last week about like translocation and important, exportant, all that stuff. So exportant is what actually helps bring the pre-miRNA out into the cytosol to get further processed. So once the pre-miRNA is out into the cytosol, it goes through dicer to continue processing. These are like the second cuts. Once the pre-miRNAs go through dicer, they now become miRNA duplexes, which is just like these little small pieces. What I said before about like 20, 25 base pairs long. And so miRNA, once they become these small little duplexes, they are ready to bind to the RISC protein. And with the RISC protein, what it does is it will unwind the double strand duplex and choose only one of the miRNA strands to use as a guide strand that it wants. The other half or the other strand that it doesn't want, it will usually be recycled back in the cell because cells don't like to waste anything. They like to conserve as much energy as possible. So it might be degraded, but usually it will just go back to be recycled and reused. And then the last step or so, once RISC has unwinded and chosen its guide strand, that guide strand acts as the complementary base pair for whatever target mRNA it wants. And the end result is usually translational suppression or being sequestered and located off to P bodies where it can also be degraded or honestly just stored there. And the reason why this is usually the outcome and not just degrade and chop up the mRNA, the target mRNA that you want, it's because miRNAs usually don't have an exact base pair. Because it doesn't have an exact base pair, there's this enzyme inside RISC, which is called Argonaute, and Argonaute will only be activated if there's a perfect target binding or a target match. But since there is not usually a perfect exact base pair match, it will just be located off to the P bodies where the mRNAs are just like decayed or stored and just, yeah, sorry. But yeah, they just come off to the P bodies to be stored away. But was there any questions about the mRNA pathways before I go into the siRNA? Alrighty. So siRNA, they're really similar to miRNAs. They're both transcriptional regulators. They act as a guide strand for RISC. It also regulates the mRNA stability, pretty small, 20, 25 base pairs, double-stranded. However, a big difference is that, like I said before, they are exogenous. So the RNA is brought in from somewhere outside of the cell. And it's usually viral. In theory, I guess it could be anything outside of the cell from like other cells, I suppose. But usually they're viral, they're viruses. And so siRNA is really important for protecting organisms against any foreign invaders, anything else that produces a double-stranded RNA that it does not want inside. So yeah, it can help suppress viral infections. And then another huge difference is that siRNA, unlike miRNAs, are usually an exact base pairing match. And then that is what will allow the end result of siRNA pathway to usually result in the mRNA being completely degraded or chopped up. So for the siRNA pathway, it actually starts in the cytoplasm. So because it is exogenous, it's coming from somewhere outside of that cell. It will not start in the nucleus because siRNA is not coded for in the genome. So instead, it'll start in the cytoplasm, in which the double-stranded RNA or the short RNA is going to be cleaved by dicer, which is the RNA nuclease that we talked about before. And then once it becomes an siRNA duplex, just this shorter version of siRNA, RISC will come in to unwind the double strand and pick one of the guide strands with the help of Argonaute. And then risk siRNA complex, this entire formation forms. And then that is when the complex as a whole will start base pairing with mRNA. And then, like I said before, siRNA is really good at exact base pair matching. So because there is this exact base pair matching, Argonaut, which is the enzymatic portion of RISC, it gets activated and it will go ahead and cleave the mRNAs, and which will cause degradation. Or obviously, once you degrade the mRNA, the mRNA will be silenced. And so that's one of the biggest takeaways. If you remember anything, it's that siRNA will has that exact base pair matching. So usually the end result is degrading, cleavage, and silencing, whereas miRNA is going to likely just result in the storing away of the mRNA in like the P bodies and such. Oh, and then something else to remember is that siRNAs, they are pretty abundant and they are really versatile too. So because of this exact base pair matching, one siRNA guide strand can cleave many target mRNAs. So if you had a lot of different target mRNAs that you wanted to take out or silence, you don't need a whole lot of siRNAs to go and target each individual mRNA. If you have just one and that one works fine, usually the cell can reuse and recycle that guide strand to continue using. And then just to drive home a little bit the difference between these two, because I know sometimes some people get confused. So miRNA starts in the nucleus. This like gray part is the nucleus. So it's endogenous, it starts in the nucleus, whereas siRNA starts in the cytoplasm. And then because miRNA starts inside the nucleus, it has drosha processing. It goes through drosha, whereas siRNA will not have drosha since it has no need to be inside the nucleus. And then again, miRNA usually does not have exact base pair matching with the target. And so the result is usually translational suppression or P-body localization for silencing. Whereas you have exact base pair matching, which will lead to the target mRNA activating the Argonaut subunit inside RISC, and then that will cleave the mRNA, and then mRNA will be silenced, degraded, not useful anymore. But yeah, those are the two main differences. Any questions about any of those two pathways? I talk a little fast sometimes, sorry. So if I like didn't explain anything clear, let me know. Okay. Well, that was pretty much it for the second half of this review session or of this week's lectures. Thank you guys for coming. Feel free to reach out to either of us or any of the TAs really through email if you have any questions. But yeah, like we're gonna be here for a couple minutes if you wanna ask questions. If not, have a good night and have a nice weekend, guys.
Bye, everyone.
Thank you.